I'm going to give a brief overview of an ongoing project that we've been working on, and you should note the numerous names beside my name on this title slide, many of whom have degrees in veterinary medicine and expertise in wildlife health, so they're driving a lot of the health part of this project. And it is an ongoing project, so I don't have any large conclusions or final results or even any detailed analysis to show you. So, okay, so Jason already showed this graph that was from Dave Hervey et al.'s paper that came out in 2013 showing those relative declines in caribou herd population sizes in all the herds in Alberta. So we know from those repeated counts that there are precipitous declines in a lot of these herds in Alberta over the last 20 years. The only one that in fact is kind of slightly stable-ish is the little smoky. I don't know if you can see that one kind of on the right-hand side. You can see it's kind of leveling off and going up. It's number 8. And as we've heard many times already today, also we know why these caribou are declining. It's pretty well accepted that it is because of this apparent competition issue, the increase in early sewer stage habitat, increase in ungulates, increase in predators. However, as we've just heard, it also is important to continue to remember scale when we're looking at research questions. So one of the questions that we're looking at for this project is looking at apparent competition from another scale. So specifically, we have this increase in abundance and distribution of moose, deer, and elk within caribou ranges. So that probably corresponds to an increase in the potential for disease transmission from those alternate prey to caribou. And this potential for disease transmission is only expected to increase with climate change. So warmer climates will allow these pathogens to survive in the environment for longer periods of time. That was one question that we wanted to try and get at a little bit with this project. Obviously it's a huge topic, but we wanted to try and get some baseline data so that we could monitor changes into the future. And also there's been a lot of focus on the big bad wolf, and obviously it is a predator that is a major driver in caribou declines, and management actions are addressing that, but particularly in West Central Alberta, there are a few other predators we need to consider. And along with that increase in alternate prey, specifically deer, we're getting a lot of increase in other predators within the system. So we have black bears and grizzly bears, but we're getting a lot more cougars as well. So we wanted to understand what role cougars were playing in caribou mortalities. So I'm briefly going to touch on both of those topics here. So just remember, still ongoing research. So our project goals, very small questions: where and why are caribou dying? And we also wanted to collect information on health baselines within these caribou herds. So there really hasn't been a lot of focus on caribou health in Alberta, but partly funded by the BC Oil and Gas Research Innovation Society in BC, there's been a recent push to collect health information on caribou in British Columbia. And because of our collaboration with those researchers, we decided to try and start collecting the same data from caribou herds in Alberta. We're not going to know if things change if we don't have any baseline data to track those changes. And we also wanted to figure out what predators are involved in adult female caribou mortalities within our study area. So we wanted to see whether we should consider additional predators when it comes to management decisions, and particularly those predators that are active all year round, like cougars. And for this project, our study area is focused in the 4 West Central herds that I talked about earlier, so we're not going up into the Chinchaga for this research. It's too far. Okay, so we're approaching our questions using 3 different approaches. We have field investigations. I rarely get to go out into the field, but this is one time that I do. We're doing monitoring of disease and health, both via field sample collection from mortalities and non-invasive fecal collections. And there's a lot of laboratory work underway in the University of Calgary and Toronto Zoo that I don't understand, but that is amazing, and they give us results. And I try and understand what the results mean. And ultimately, when we get all these data together, we're going to try and do spatial modeling of disease and differences amongst herds. It really depends how much data we get. The lab results aren't in yet. and also look at potentially mortality risk in relation to anthropogenic disturbances and predator overlap as well. So it's building on the spatiotemporal analysis, and specifically for the mortality and predation risk is building on a lot of existing research that was carried out by Nick de Caesar during his PhD, and also that Gord has been carried out with the grizzly bear program funded by PTAC last year actually. So field investigations, what do we do? Well, these collars, our collars worked, Jason, and they send us an email if the caribou doesn't move for 8 hours. I could have done a graph, but these emails are usually sent on long weekends when I'm on vacation and when I have to be elsewhere for meetings. So it is a strong and horrible correlation, but what we do is we get these emails and we try and get out to these mortality locations that day. If we're lucky, it does happen that day. Sometimes, depending on weather, it cannot happen that day. But we have a really good helicopter company that we work with that's willing to just drop everything and come out with us. And my biologists are becoming used to the fact that they have to drop everything, no matter what day it is. So yeah, we get to a lot of them in recent years within 2 days, but some of them are a week. And then sometimes in the winter, if it's really windy, we've had a lot of warm winters the last few years, and the winds getting into the mountains can stop us, but we try our very best. And because we're getting to these mortality sites really quickly, we're getting accurate predator ID. So we're finding that the longer it takes for you to get to mortality site, the harder it is to tell what predator was involved in the mortality. So we've had some where we went to the mortality site 3 days after she died and it was a cougar, but we didn't land. We just recorded it and went home, and then we came back a week later and all the signs were grizzly bear. So that was a cougar predation event, but scavenging by grizzly bear. But if we'd just gone and like a week later, then we would have classified that as grizzly bear. So it's really important to get to these sites very rapidly. And also, if you get to them rapidly, you have a lot more samples to do these disease and health testing on, and you can really get accurate predator ID, even if you don't get a predator visual, and there might be enough left to you to figure out what predator was involved in the caribou's death. And yeah, we try and do detailed necropsies in the field and collect as many samples as possible. Sometimes we'll bring the carcass or partial carcass back to Hinton and do a detailed necropsy there. Again, our helicopter company that we work with is very obliging with that. Okay, so depending when we get there, there can be different things left at the site, and we're really relying on expertise in the field. We work a lot with the grizzly bear program, and we'll even send pictures around to wildlife biologists in the government of Alberta to try and figure out, okay, who do you think, what predator was involved in this mortality? But depending what was there at the mortality site, it can sometimes be difficult to tell. And so this is a picture from what we classified as a grizzly bear site, but because we got to this location 2 weeks after she died, we couldn't classify this as scavenging versus predation. So that's another hard thing to say. We know when the caribou dies, but we don't have the predator collars. We don't know when the predator got there. So we do these detailed site investigations, expert opinion, but it's very hard usually to tell if her heart was beating when the predator got there. So yeah, we were coming up with a confidence criteria for our data set. So probable predation, probable scavenging, definite predation, depending on what was there and how quickly we got there. This is showing a bear scat with lots of caribou hair in there. So we know that bear ate her, but again, was the caribou alive when the bear ate her? This was quite a lucky find, actually. It was actually the first mortality we ever went to, and we didn't get to her till 2 weeks after she died, but there was enough left of her carcass that we could figure out predator ID. So this is showing, you can see the marks in between, so there's 3 holes in her hide, and there's 2 marks on either side for canine marks, which we classified as bear because of the distance between the canine marks. And on the underside of the hide, there was darker areas around those canine marks, which are ante-mortem hemorrhage. So her heart was beating when the bite was made. So that one we were lucky enough to be able to say, okay, bear predation. But finding that sort of data at a mortality site, particularly 2 weeks after, is rare. It's like gold. But We're collecting data gradually, but good important data to have. This is one, anyone guess the predator? It's a wolf. So this is imagined, so on the right hand side there's the two canines, and then going off in the V are the animal's molars and premolars. So it's kind of the indentation of the wolf's jaw. So this is one that we kind of missed in the field because it was -10 when we went to collect the carcass, but the vets pointed out to us afterwards. But this one you can see there's kind of dark stain around this hole in the hide. So again, her heart was beating when that bite was made. So that was a wolf predation event. So yeah, just a few examples. But sometimes, no matter how hard we get there, it's just pretty confusing. So this is one we got to the day that she died, and all we could conclude was she fell from the cliffs. This is Mount De Beaver in the Wilmore, and this is the mountain. It's 800 meters high on the left. I don't know if you can see the faint blood trail coming from the base of the mountain. So this one we classified as accident, but was she chased or did she fall? We couldn't figure that out. It was too windy to circle around the other side of the mountain, but we did classify her as an accident. So there's a lot of things against these animals, gravity included. So this is what we got. So what we did is we actually started this project in 2013, funded by Weyerhaeuser, and then PTAC were generous enough to, or Alberta Upstream were generous enough to support it last year and this year. So we have 3 years of data. And what you can see from this table is, first, there's not a lot of data in this table. But if you remember that each line on in this table represents a dead, threatened or endangered adult female, then it's probably not a bad thing there aren't a lot of lines on this table, but from a statistical point of view, it's not ideal. What you'll also notice is it's really detailed and it's really hard to read. So, in summary, what we got from our 3 years of data collection going to these mortality sites, we had 12 caribou that we classified as predation events based on our field investigations, 4 of which are cougar, 2 are wolf, 3 grizzly bear, and 3 have multiple predator signs that we couldn't really figure out if we'd done it. Of the remainder, 3 were accidents. So one was that one that fell off the cliff. 2 were road traffic collisions on Highway 40. The Alberta animals migrate across Highway 40 during the spring and fall, and sometimes they get hit by vehicles. We had a male in 2013, a young male in 2013, an older male in 2014, none last year. And unfortunately on Tuesday we had a prime adult female killed by a road collision with a vehicle on the highway. 3 looked like they just dropped dead. We don't know what killed them. So we're classifying that as disease ? But it's really hard to kind of, depending, like some of these carcasses were a little decomposed when we got to them, but we're doing as much disease and health testing as we can to try and figure out why they might have died. And 3 were unknown. There just wasn't enough left for us to figure it out. So yeah, that's our huge data set. So because we don't have a large amount of data, we are augmenting our data sets by using non-invasive winter fecal collections. So as I said before, we do these detailed necropsies back at the office or in the field to collect as much detailed health data as we can, and we get our wildlife veterinarian. He lives in Calgary, and he'll drive up at the drop of a hat and do a necropsy on a caribou. He was up yesterday doing the road-killed one. But we are getting additional data from winter fecal collections. So if we've heard about before, non-invasive sampling of wildlife is increasingly giving us a lot of information. So what we are doing is we identify individuals using genetics. So that takes about 5 or 6 months in the lab. And then once we have the individuals identified, we send the samples to Toronto Zoo, where they do pregnancy testing and stress hormones. And they also go to the University of Calgary, the Canadian Wildlife Health Cooperative, where they measure pathogens within these species as well. So we're getting a lot of information from our winter fecal collections. And what we're getting, so primarily we're looking at bacteria. So depending on what's left to the mortality site, we can't really do a lot of very detailed kind of looking at organ sections and so on. But one thing that we can get is we can test for bacteria within the bone marrow. So if there's even one intact long bone or an intact lower jaw at a mortality site, we can culture bacteria from the bone marrow in that bone. So that's a pretty consistent sample, pretty consistent thing that we can test for across all our mortalities. We're looking at winter fecal pathogens. Winter tick is another thing that we're looking at. It's quite common in moose. It could be transmitted to caribou. And then all these other health metrics like stress, bone marrow, and trace nutrients, all indicators of just general health. Okay, so very preliminary results based on a very small data set, so ongoing project. Bacteria-wise, one of the main bacteria we're looking at is Erycephalothrix. Don't ask me to pronounce the second part of its scientific name. We identified it in 6 of our 11 mortalities. It's also been, and it's that bacteria you can get from the bone marrow. It's been identified in caribou and moose in BC as well. And it's been associated with mass die-offs of muskox in the Northwest Territories. Its role in caribou mortalities or disease, we don't know yet. It's an emerging pathogen. The University of Calgary has a research group looking at it in detail. We don't know if it's causing deaths or if it's causing mortalities or if it's simply something that's exacerbated by other conditions that the caribou encounters, like increased stress or something like that. So to be continued with respect to the importance of this bacteria for caribou. We're just looking at it right now. Fecal pathogens, we've only 1 year of data so far. We found a few of the usual suspects in the feces, but we don't have enough to do any detailed analysis. We detected winter tick on 3 of the animals that we tested, one of whom was severely anemic. She had a lot of winter tick on her. And bone marrow fat, 3 caribou looked to be had relatively low bone marrow fat, and one was classified as extremely low, and this was the caribou that also had that heavy infestation of winter tick. So hopefully I'll have more detailed results to show you next year. So yes, from our year 1 of data, what we found is going to these mortality sites, there's a range of different pressures involved with adult female caribou mortalities in West Central Alberta. And we are collecting this baseline health data on pathogens and bacteria and disease that could be used to track changes over time. Ongoing work, we're going to continue to visit these caribou mortalities. At some point we're going to have to stop collecting data and stop analysing it, but when the data is so rare it's hard to say stop. But regardless, we would like to continue this project. We'd like to continue visiting these mortalities and getting this valuable health data. We should have the lab results from Toronto Zoo and University of Calgary pretty soon. So we'll get to look at those, and hopefully we'll have enough to compare amongst herds and across the winter. And what we want to do as well is we want to create maps of mortality risk for caribou. So we want to, so this is building on work that Nick DeCesar did, and there's similar work is being completed for grizzly bears. We want to look at the locations of caribou mortalities relative to anthropogenic disturbance and different habitat layers, and then as well as with overlap with different predators. So Nick DeCesar did it for wolves, and the grizzly bear program did overlap between grizzly bears and caribou during the calving season last year as part of their PTAC project, but we want to expand it to include cougars as well. And then we'll integrate that with our field investigations. So our caribou dying from cougars in areas that are really preferred by cougars or marginally preferred by cougars. So hopefully all of this will feed into landscape management and helping to figure out where we need to restore caribou habitat, because we want to keep caribou around. Maybe we want to target restoration further to broader areas where there's a high mortality risk for caribou, or where there's a high predation risk from different predators. With respect to disease and the management applications, they're not as direct, but we do have a complementary project looking at disease in ungulates within caribou ranges. So hopefully with this baseline data on caribou and ungulates, we'll be able to identify herds that could be at risk from disease outbreak and perhaps make management decisions to reduce the potential for disease transmission between moose, deer, and elk and caribou. And of course, we hopefully will have enough data to track changes over time. It'd be really nice if we could, again, do this sampling maybe 5 years from now and see how things have changed as we implement restoration on the ground. So that's kind of it. A lot of collaborators in this project, FRI Grizzly Bear Program, Alberta Agriculture and Forestry and Environment and Parks, and all the different labs, and as well as a large number of funders and in-kind support.